presentation is by Dr. Varshney. Well, as you all know, the organizer of this particular meeting, but otherwise also the molecular breeding, in the area of molecular breeding, Dr. Varshney's contribution has been monumental, and I'm sure a number of breeders, especially the pulp breeders in this country, are taking advantage of his expertise. I'm sure he's going to integrate both the tools as well as the traits. Both are important and they are needed in integrated plant breeding. Thank you. Thank you, Dr. Varshney.
Thank you very much, sir. Some of you must be surprised at how I became so tall, right? Because my colleagues are great. They told me yesterday that I was invisible behind this podium, so today they organized the platform. I wanted to perform on this platform, they made me stand on the other platform. Anyway, thanks guys. So now I'm going to speak on this integrated breeding in grain legumes, and what we will do that we'll give some examples from 3 legume crops on which we work at ICRISAT: chickpea, pigeonpea and groundnut. Before I present, I would like to mention that this is a contribution from several colleagues. Majority of our breeders from ICRISAT, physiologists, biometrics, bioinformatics, genomic scientists, and of course several collaborators who are sitting in this room. So this is basically contribution from different colleagues. So thank you very much for collaborating with us, and I feel privileged to work with so many people. What I'm going to do in this talk that will give very briefly some idea about those legume crops on which we are working, and then our efforts on development of genomic resources, because 5-6 years back when I started to work on these crops, we did not have even enough markers. For instance, in the case of pigeonpea, we had just 10 microsatellite markers when I joined ICRISAT, so those were the thing. But anyway, so then now, but in collaboration with colleagues from ICRISAT and from outside, we have developed, I think, significant number of markers and large scale genomic resources in these legumes. I would be presenting those things. And as Dr. Sharma mentioned, so this is genomic resource development is one thing. We can publish good papers, but being, as we are working at ICRISAT, and our mandate is basically helping the breeding program. So the main thing is how we can integrate these genomic resources, what we are developing in the breeding program. And I will give some examples in chickpea, pigeonpea and groundnut. Very quickly, so we work here in these 3 crops, and chickpea is a diploid crop. We call it cool season legume crop. Pigeonpea is warm season legume crop, and genome size is about 740 megabase per in chickpea, 813 pigeonpea. Groundnut is a allotetraploid. Genome size is quite big, 2500 megabase pair. And in case of production constraint, drought is a serious problem in chickpea and groundnut, and also in pigeonpea. But there are several other problems like fusarium wilt, ascochyta blight. In case of pigeonpea, we have fusarium wilt, sterility mosaic disease. And in groundnut, we have foliar diseases like leaf rust and late leaf spot. So we are working with our colleagues to work to improve, to basically for improving that crop productivity under the target environments. So what happens, so unlike other crops, if you see the yield in the farmer's field, in chickpea, pigeonpea and groundnut, we are not having more than 1 tons per hectare. On the other hand, we have the huge yield gap, or potential yield is quite high. So the question is how we can bridge or how we can fill up this yield gap. So this is very important. So in this direction, of course, this molecular breeding or integrated breeding is one of those important approach. But as a prerequisite, you need to have the genomic resources. You need to have markers, you need to have the genetic maps, etc. We were not that privileged a few years back because we did not have those resources in these crops. So what we did, together with collaborators like BGI, and we are having Gangyun Zhang here and Wang Jun was yesterday. We have been enjoying our collaboration with BGI and also with Macrogen Generation Challenge Program and University of Georgia, UC Davis, in collaboration with those colleagues and several other partners which are not listed here right now. We decoded the genome sequence of pigeonpea, and that was published in 2011. And then after that, the same thing we did in the case of chickpea. Here we worked again in collaboration with several partners, including University of Queensland, UC Davis, Georgia, Saskatchewan, ICR, and many other partners. So we completed the genome sequencing of chickpea as well. In the case of peanut, we are partnering with in 2 initiatives. One is US-led initiative, and I think Baoju Go is going to present progress on this component. I'm not going to present further here. There is another initiative on the genome sequencing of peanut, and this is being led by China, and we are partnering there as well. So, and in this initiative, I think that sequencing work has been completed, and right now we are in process of analyzing the data. So we have completed the reference genome sequencing in all of these crops. Now, just reference genome doesn't provide everything. We had wonderful presentation yesterday, today. So if we would like to understand the genome variation, we need to work in this direction. So what we have done that at ICRISAT, we have so far sequenced about 1,000 plus genome. And in these one, we are having about 544 lines in chickpea, 526 lines in pigeonpea. In chickpea, pigeonpea, you can see that we have the reference set, parental lines of mapped population, elite lines. In the case of pigeonpea, we are having the parental lines of hybrid, because in pigeonpea, we also have hybrid system, as Jochen mentioned in wheat. In pigeonpea, we have cytoplasmic male sterility based hybrid system. So our objective is to understand those variations, to map the different traits, and to improve the parental lines of hybrid as well as helping the varietal improvement. So in some cases, data has been analyzed, and in some other cases, we are still analyzing the data. So, but again, so we have sequenced more than 1,000 pulse genomes. Now we are moving further in the different direction, and yeah, let's see. So I'm not going to present all these results, but very quickly. So you can see the beauty of the genome sequencing, and this is just result on the 90 genotype sequencing. This was the first set. We could generate more than 4.4 million SNPs. Earlier, we did not have even SSR markers. So this is the beauty. Same thing in the case of pigeonpea. If you see that this is the result based on the sequencing of 300 genotypes, we got about 9.4 million SNPs when you compare with the wild species. And when you have a comparison with the land race and breeding lines, then also you are having several million SNPs. And you can understand their frequency distribution along the chromosomes and in different genic regions. We are also in process of identification of the copy number variation in presence and absence variation, and trying to associate with the traits of our interest in which breeders are interested. But you can see from this slide that in presence absence variation, they are more in wild species as you can expect, and the copy number variations, they are more higher in the breeding lines as compared to the land races and wild. But again, these data analysis is still in progress. But before we had those genome sequences, etc., at that time point, we wanted to use the markers. In that scenario, initially we started to sequence the transcriptome, and then at that time point, we sequenced, or we used Sanger sequencing technology, 454, Illumina technology. So by using a combination of the different technology, we defined first version of the transcriptome assembly. At that time point, we had very small tags, etc. But now by combining huge data, we have a second version of the transcriptome assembly. And this was recently published, and here this is much more improved version of this assembly. But when we develop this improved version, we have combined the data from all over the world and put these efforts together. And this is very important source of for doing gene expression analysis, etc. Same thing in the case of pigeonpea. Again, in pigeonpea, we sequenced transcripts from the range of the tissues, from the different genotypes. And now we also, in this one, we are having improved version of the transcriptome assembly in the case of pigeonpea. One more thing I would like to mention, as I mentioned about this cytoplasmic male sterility based hybrid system. So what you need, you have the A line, which is male sterile. You have the V line, which is the maintainer. And when we develop the hybrid, we need to make the cross between A and R. Then we have this hybrid. And this male sterility has come from one wild species. So what we do that for maintenance, we always, for production of the seeds or so, we need to use this maintainer line. So by doing this thing, we have this very functional hybrid breeding system, and we already have several pigeonpea hybrids commercially released. And they say this is the first legume crop where you have these hybrids. So thanks to our breeders in the area of pigeonpea breeding, from pigeonpea breeding. So what we did that we sequenced those 4 lines, and which is one A, another is B, and another is hybrid, and also the wild species from where we had the cytoplasmic male sterility. So we sequenced these mitochondria from all those 4 different lines, and then try to understand. Of course, one is that gene annotation, that we try to understand what kind of genes are present, and our main purpose was to understand the rearrangements, or can we identify some variants which are associated with the cytoplasmic male sterility. So what we did recently that we targeted 34 genes, and then we analyzed those 34 genes in both ways. One is expression analysis between A and B lines, and you can see several genes which are having several fold expression variation. On the other hand, we also did this their assay on agrogel, and we found very interesting deletion, 10 base pair deletion in NAT7A gene, and then this was very diagnostic, and we could see this thing at later stage that whenever you have any impurity of the B line, this can easily be detected. But next question is that what is this A, or what kind of deletion we are having, whether what kind of genes or amino acids this is making changes. So as I was saying that if you are having ICAPA line, so those 10 base pair insertion in A line is giving some amino acids, in the B line you don't have. And when we did this analysis at the protein level, we found that there is a conformational changes, and we are predicting that maybe this is the responsible or one of the responsible factor for cytoplasmic male sterility. But again, we need to validate those results later by using different system. So now when you have the genome resources, transcriptome resources, mitochondria, breeders are not that happy. Breeders say this is good that you have published the paper, but now in the end what you can deliver to us, and this is a very important question. And as a CGR scientist, this is very important, very challenging, and we need to work in this direction. So after genomics, if you would like to use them in breeding program, then you need to have an interface, and we consider as genetics. So from genomics, you need to understand the genetics of the trait, then you move to the breeding. So to understand the genetics of the trait, you need to have the genetic maps. And what we did that again, during last few years, we have been developing different genetic maps. In the case of chickpea map, well, of course, this is the old picture, but the new map is having several thousand loci. But anyway, so we are having, so at that time point, 1200 loci, but now we have several thousand. And recently in collaboration with NIPGR from Delhi and UC Davis group led by Minching Luo, we also have developed the physical map. So recently what we did, we integrated this physical map, genetic map with the genome sequence. So now you can enter from any direction, either you have QTLs, then you can identify the genes, or if you are having some back context, etc., you can get the gene sequence, or if you are having some idea on the gene sequence, you can go to the QTL. So this is very important resource, and which will be helping a lot in coming years to the breeding community. In the case of pigeonpea, again, we had developed several intra-specific map and also inter-specific map. And in the case of groundnut, again, we have been working with different partners, and we have developed first a high density map, and after that we merged different maps. And tetraploid peanut marker polymorphism is very, very low. So if you are having maps of 3000 loci in wheat, rice or maize, they are not that important. But in the case of peanut, if you have a genetic map with 3000 loci, this is highly, highly important map for the breeding applications. So then when we have the genetic maps, and then we wanted to understand the genetics of the traits. And as I told that in chickpea, drought tolerance is one of that important trait for breeders. And drought is very complex. So in this case, our physiologists like Kashi Wagi, L. Krishnamurthy, Vincent Wadage, etc., and together working with breeders and genetic resources specialist people, they concluded that root traits, and when I say root traits, this is not one, this is basically the suite of the traits, root land density, root biomass, root shoot ratio, etc. They're very important trait for conferring drought tolerance. Breeders say this is fine, but we would like to see the yield or harvest index in rain-fed conditions. So what we did that we, Puran Gaur, who's our chickpea breeder, who developed two fantastic mapping populations, one is ICC 4958 and 182, another is ICC 283 and 8261, there is some problem with formatting. But anyway, these are the two intra-specific mapping populations, and what we did, that we did extensive phenotyping for 3-4 years on a range of the traits, all kind of traits which you can imagine, including transpiration efficiency, etc., carbon isotope discrimination, and in different environments. And after that, we did this genetic analysis, and we found a genomic region in this population, which, and I call it QTL hotspot, because this QTL hotspot is having the QTLs for more than 30 drought tolerance related traits, and in several cases, the phenotypic variation is going up to 58%. Interesting thing is that this is not dependent on the donor genotype. When we have this other population where we are having the different set of the genotypes, we found the same genomic region here. So basically, this gives us a lot of confidence that maybe this genomic region having some interesting genes. Maybe they are not directly related to drought tolerance, but maybe some transcription factor which are controlling the expression of the genes which are associated with drought tolerance. So at that time point, this genomic region was quite big, 20 centimorgan or so, but recently we have undertaken this genotyping by sequencing approach on this mapping population, where we sequenced each RIL at 0.38x coverage, and then we have put more than 30 or 40 markers of those SNPs in this region, and then we are working further that how to move ahead. Next thing is, so this was linkage mapping approach for mapping the drought tolerance in chickpea. We used other approach, this is association mapping. In association mapping, Dr. Upadhyay from our gene bank, he developed a reference set in collaboration with Generation Challenge program. And what we did, and of course in collaboration with ICRISAT, in chickpea we have 20,000 accessions. These guys, they came up with a set of 3,000 accessions. They said this is global composite collection. Initially, we did the SSR-based diversity analysis on these 3,000, and then we developed a smaller set, and that was 300 genotypes, which is not having very strong population structure. So that set is very ideal set for association mapping. So what we have been doing in past or during last few years, and NJ Killian is sitting here, that with DART company, we also developed the DART arrays in all of these three species. So thanks, Andre, for that one. And what we did that then we used those 15,360 features DART array to genotype all these sets, but you can see that level of polymorphism is very low. But anyway, we had this marker data for 1,843. In parallel, we also have some candidate genes, and then we also sequenced entire reference set for those candidate genes. And then what we did that we combined all these data. And again, this is a different set of the mapping population, and this reference set was also phenotyped several locations in India and in Ethiopia and in Kenya. And then we combined all these analysis data and we found several peaks. But most important peak is that this is carbon isotope discrimination and yield under rain-fed conditions. And when you see their location, they are going in the same QTL region which was identified through linkage mapping. So this enhanced our confidence further that this genomic region is having some interesting stuff to target. Now we also did, well, of course, the GWAS results at that time point based on markers. But now once we have the genome sequence data at the level of 5x to 15x coverage, we are moving in the direction of the GWAS analysis at the genome scale at that single nucleotide level. But at the level of the markers also we got several high peaks for root length density, 100 seed weight, harvest index, etc. And of course, when you do the candidate gene analysis, then we found several SNPs in different genes which were found associated with this drought tolerance. And then now, because we know the geographic distribution of the different lines from where they are coming, we have very clear idea about the haplotype distribution of those genes or alleles which are associated with drought tolerance. So this is going to be very useful information. Then in the case of chickpea, again we are having some problem, fusarium wilt and ascochyta blight. This project is a part of accelerated crop improvement program which is being led by Professor Gupta. So what we did that we also tried to do the QTL mapping in these for these traits, because earlier QTLs or genes were mapped, they were mapped from the different genetic background. So this was a bit complicated stuff at that time. But now we have mapped the fusarium wilt and ascochyta blight also in the case of chickpea. In the case of pigeonpea, fertility restoration is another important thing. And now my colleagues, they have developed a mapping population. They did some preliminary work, but these results are not that exciting, but we are working further in this direction. Recently, we have used this approach called QTL-seq, which Rohit Rauchi has optimized or presented yesterday also. So what we are doing here that we have those two parental lines, and then these parental lines, they segregate for fusarium wilt and sterility mosaic. And then we had this RIL data, and then based on the phenotyping on data on the RILs, we developed two different worlds. One world is resistant, one world is susceptible. In the first instance, we took 16 plants from each world, and we did the sequencing of more than 8x coverage. And then by analyzing this SNP indexing method, you can identify that these peaks, so that for instance, here when you see the variation between this bulk for resistant and bulk for susceptible, you can easily land on the SNPs or the linkage group. So for instance, in this case, we found one SNP for the SMD, here linkage group 2. And interestingly, when we compared this thing with our previous results, we also had one QTL in this direction. But anyway, so now by using this approach, and we are applying this approach at large scale now, but by using this approach, we identified about 14 SNPs at different linkage groups. And now based on some bioinformatic analysis, we are narrowing down this list further and also sequencing those genes across that set of that diverse germplasm, etc., to see whether we can use these SNPs as a diagnostic markers for fusarium wilt and sterility mosaic. That was about pigeonpea. In the case of groundnut, as I told earlier, that late leaf spot and rust, they are important constraints. So what we did that together with the group from Dr. Chanvara Gowda, at that time he was at University of Agricultural Sciences, Dharwad, by using mapping population which he developed, we identified several QTL regions, but there is one region, this had the major QTL for rust, and this contribute up to 80% of the phenotypic variations, and we also validated this thing in different genetic background. So what I would like to mention that after developing the genomic resources by using the different mapping population and phenotyping data from the different colleagues, we had fair understanding of the genetics on the different traits. So the next question is to work in the breeding program. And then for the drought tolerance, and as I mentioned the name of Puran Gaur, so generally as a academic scientist, majority of time what we do, that you have drought tolerant QTL, you ask people, give me the drought sensitive line, I will put this drought QTL in drought sensitive line, will make your line drought tolerant. Puran Gaur is very pragmatic breeder. He says, Rajeev, I don't work on this drought sensitive line. If your QTL is having some potential or some value, I will give you the line JG11, which is the most drought tolerant line in India and widely grown in Andhra Pradesh state, Karnataka state, etc. He told, transfer this QTL in JG11. And then we will see how much impact or how much yield this QTL will add. So this was very challenging. So what we did, that you can see, as I was saying earlier, so again, don't take the wrong message that roots alone is everything, but I'm giving this as example. So JG11, which is high, they say very drought tolerant variety, and ICC 4958, where we identified the QTL, you can see ICC 4958 is having higher rootland density. If you are having the traditional chickpea line, there will be less, but JG11 is already drought tolerant. You can see this is quite high. But by doing these 3 back crosses and by using these markers, which we had at that time point with us, SSR markers, we developed several back cross progenies, and now you can see that several back cross lines, they are having higher rootland density, of course, better than JG11, but in several cases more than ICC 4958. So we have confirmed this thing that we could introgress this trait in the breeding, in the elite cultivar. Again, breeders are not very happy. They say, so what kind of yield you are having here? And this is very important. So what we did, that we have done this multi-location field trial, 3 locations in Africa, 3 locations in India, and our preliminary, I'm using the word preliminary. So preliminary results are very exciting. Now we are having several lines that when you have this irrigated conditions field trial, we are having 12% higher yield than the elite. And this 12%, I'm talking on the topmost or most drought tolerant variety. If you would just transfer this thing in the drought sensitive or local normal varieties, maybe this gain will be quite high. But anyway, but another thing is that if you are having, if you have the rain-fed condition, 12% gain. But if you are having irrigated conditions, then this gain is 24%. So this is very interesting. And yeah, so this was also cover story in the plant genome. So we already, this is Puran Gaur, many of you might have already met him. So now what we are doing, that we are doing several things. One thing is that in collaboration with Generation Challenge Program, Bill and Melinda Gates Foundation, we are or in collaboration with Department of Biotechnology, Government of India, our plan is, can we introgress this QTL and other chickpea varieties to see how well this will do. So now what we did, that together with several partners from India, Ethiopia and Kenya, we have developed several backcross progenies in different genetic background, and right now they are at different stages. Another thing is that because we had those we had these backcross progenies, we also developed the near isogenic line. So now we are having one near isogenic line, we call it JG11 plus, where we have a QTL from ICC 4958. We are also understanding in 2 things, to do the QTL cloning, and another is to understand the genes that which are giving drought tolerance. And for this one, we did this MACE expression analysis by using RNA after imposing the drought on these genotype. And we find 27 differentially expressed genes, and they are very interesting genes. You can see that in ICC 4958, you have this expression in 182, drought sensitive, you don't have anything. In JG11, good. But when you have the JG11 plus, this is going quite high. So this means that this gene is having some interesting thing to play with JG11, and we are identifying a lot of stuff. Now some of my colleagues, they are again interested to understand this protein-protein interaction, and they are trying to predict this. Maybe my one RN1 is involved in the drought stress. So we are working in this direction. That was the story for the drought, which is very complex trait, but we have done more or less similar kind of thing in Fujiram wilt and Ascochyta blight. Here you can see for the Ascochyta blight line, so we are having this is susceptible check where we transfer this Ascochyta blight. You don't have anything, but when you are having this QTL, you got those lines resistant. The same thing we are having for the Fujiram wilt, that when we have this susceptible check, your plants are susceptible, but when you introgress these QTL, you are having sometimes 95% resistance, but in several lines, you are having 100% resistance. In the case of groundnut, I told that we had a QTL for late leaf spot and leaf rust, so we use the same approach, marker assisted back crossing approach, And this QTL came from GPVD4, ICGV 9114 is high, this drought tolerant variety, but became susceptible for rust. What we did, we transferred the QTL from here to here. JL24 is the susceptible check. We also transferred QTL from here to here, like that. And now we can see that just by introgressing one QTL, you can see the good symptoms when you have the introgression lines. And my colleague Janila is doing wonderful work together with Manish Pandey and Dr. Upadhyay. And here you can see the tag 24, which is a susceptible parent. You don't have much yield, but so this is also impacting on the yield. And again, so these yield trial, this is undergoing, and we will see what will happen. Now, in the case of peanut, in collaboration with Mars company, we are now trying to pyramid rust resistance, late leaf spot, and high oleate trait. I have taken this slide from Roberto Tobrosa. Probably he will not present this slide tomorrow in his presentation. Sorry about that, Roberto. Okay, good. So anyway, so the point is that whatever we have been doing so far, we are using this MACE or MABC approach, and Roberto says this thing, that so far we have been touching the tip of the iceberg, but huge stuff is left here. So we need to target this thing, especially when you are targeting or when you are talking these traits like drought tolerance or yield under target environment conditions. So in this direction, we are having some collaboration with Cornell University, John Lukyanik, University of Edinburgh, John Hickey, or University of Nebraska, Aaron Lawrence, and several other colleagues, that we are trying to understand this phenomena of genomic selection. We started to use this genomic selection in our two crops, chickpea and pigeonpea. And what we are doing that in chickpea, we have a training population. We are also having a diverse set 309, but as Jochen mentioned, so sometimes when you use the training population, if they are very far from that breeding population, they are not very useful for prediction. But this training population is coming from that same germ, from the germplasm, which is being used in the breeding program. Nevertheless, we are also using some Y parental population. The same thing, we are having this different set of the population. So right now what we are doing, that we have compiled the phenotyping data on these population. In some population we had the historical phenotyping data, so we compiled that one for last 10 years. In parallel, in the case of chickpea, for instance, recently we have generated additional or new phenotyping data for 2 locations in India, and we also did this genotyping with Casper markers, DART-seq, and then by using these data, my colleague Abhishek Rathore and other colleagues, we have been working this thing that, and also with our collaborators to, first we are doing 3 things. One is we are trying to understand or compare, we are comparing the different genomic selection model. Second number is that which population will be good, and third is that which traits we need to target for the genomic selection. So for instance, and in some of our results, we were very exciting results, that our prediction accuracies for genomic estimated breeding values, they were in the range of 0.0443 to 0.99, which is quite high. Cross validations also went very nicely. So then this is again varying from the trait to trait. So in addition to that one, so now our objective is basically to working with the national partners. So in collaboration with Generation Challenge program, we are developing a pipeline for genomic selection. We called it ISMU version 2. And again, this in collaboration with John Hickey, Aaron Lawrence, and John Lukyanik, and some of those information is present on the poster. So idea is that once you have the huge amount of genotyping data, and as Richard mentioned in morning, that many times breeders or geneticists, they are not expert in bioinformatics, it's difficult to analyze this data. So if you have the pipeline, you feed this data, and then as a breeder, you can just press the button. In the end, you would like to know which line I need to use for making the crosses. So we are working in this direction. In the end, the take-home message is that genomic resources, in my opinion, is no more an issue in these legume crops now. And molecular breeding for simple trait is in routine. I already have given some examples. Phenotyping, when you are targeting the complex trait, we need to have cost-effective and precise. Cost-effective is also important thing here. Development and use of appropriate decision support tools, and then this is happening, and colleagues like Gary Atlin, they are, and many other colleagues, they are having some initiatives. We would like to work with them, that how to contribute in this direction, and then after that, deploy those tools in our breeding program. And in our opinion, genomic selection needs more attention. And in India, sometimes people say that genomic selection is the approach of future. I say, no, you need to start from right now. So of course, you may not have perfect model right now, but you should not wait for perfection. We will be improving ourselves in parallel. And then the last, that we believe in open data access and empowering national programs in modern breeding. And again, so in our center of excellence in genomics, we are collaborating with more than 110 organizations, and majority of them, they are here from more than 35 countries, and we feel very privileged about that. So thanks a lot for that, and I would be happy to answer any question.
Thank you very much, Rajeev. In the essence of time, let's move on to the next session, and we'll take
I still have 1 minute, so
Okay, then one question. One question.
Okay. No, in fact, I'm nervous as well, so I don't want to answer any question now.
We can take the questions at the end of the session. Pardon me? Yes, sir.
Okay.
So let's take the questions at the end of the session or during coffee.
Thank you.
Thank you, Rajeev.
Yeah. Maybe I can move this plate from now. Right?